Hi, in this video I am going to talk about the bioprospecting of antiviral agents. I don't know how to explain the importance of the antiviral agents, especially in this COVID pandemic period. So the antiviral agents can be found from nature, and most of the antiviral agents we are using now is actually found from natural resources. So virus cause serious diseases, and other thing is that it may produce novel serotypes like the COVID-19. So the coronavirus, new novel coronavirus is a new serotype of the coronavirus. So this thing can complicate things and make highly pathogenic infection. The antiviral drugs, you already know the biology of the viruses. Virus is actually using the host cell machinery for the growth and multiplication, replication, etc. So the antiviral drugs can be either use the viral proteins or viral components as the target, or it can use the host cell factors which is used by the virus as the target. So two different ways it can act. And the cultivation of virus is very much different than the cultivation of any other cells, because viruses are growing in living cells. If you want to culture or grow a virus which is infecting bacteria, what we need to do, you need to grow the the bacteria first, then the virus will be inoculated on top of the bacteria. If you want to culture a human virus, first we need to culture the human cell, and to the human cell culture we'll be inoculating the virus, because the substrate for virus is the living cells, so it needs presence of living cells for growth and multiplication. Or we can inject it directly into the organism. If the virus is infecting the rat, go and inject into the rat. Or we can use the embryonated eggs. Many of the animal viruses can be cultured in eggs, the embryonated eggs. So we have the in vitro and in vivo cultivation. In vivo cultivation we are using the whole living organism or the embryonated egg. In vitro culture we are using cell culture, which is the most common method. So the antiviral agents has different targets in viral life cycle. Either it can prevent the absorption and penetration, then or it can prevent the uncoating, the DNA release into the cytoplasm can be prevented, or reverse transcription, like the transcription or reverse transcription can be prevented, or the protease inhibition, which is an important step in most of the virus life cycle, or we can actually prevent the release of the virus. So this is a typical example of retrovirus. That's why, but most of the viruses has same point of inhibitions. So what the whole meaning of bioprospecting of antiviral agents? This thing, actually the bioprospecting cannot be done in animals. We cannot actually take the animals infected with some viral diseases and inject the natural products for cure. We are actually doing it in cell culture system. The animal cells can be cultivated in flask like this. This is called a tea flask. I think you might have studied about it in your plus two or something like that. So the animal cells can be cultured in this type of tea flask. It has a rectangular medium, and in its bottom the cells will grow like this, just like the monolayer. Okay, this one is uninfected. If you add viruses into this medium, what happens is like the viruses go and infect the cells, and eventually it will kill the cells. So here you can see the healthy cells, the elongated cells, the fibroblast like symmetric cells, but here everything is round up. The round up cells actually indicating the dead cells. So if you infect the virus cell culture with the virus, the cells will die, and the type of death will vary depending on the type of cell and type of virus, but basically this is what is happening. If you infect the animal cell culture with the virus, it will kill our cells. And if you want to study the effect of some plant extract or some animal proteins against this virus, what we need to do, along with this virus, you need to add the compound of interest, or the compound you think is antiviral. So if the compound is able to prevent the cell death, if you are adding virus along with this compound, actually if you are adding virus it should be look like this, but if you are adding compound along with the virus, if you see something like this, like just like the uninfected cell, your compound is doing something to the virus which prevent the infection, which may have a clinical application for the treatment of the disease. So that's the way we are actually doing the bioprospecting on the screening of antiviral agents. For the screening of antiviral agents, we have different methods. I am pointing out some of the most important methods for bioprospecting of antiviral agents. First one is plate inhibition assay. Plate inhibition assay is also called plate reduction neutralization test, PRNT. In this, what we are doing, we will take, just like here, we are taking plates, and this plate, we are actually putting the viruses in it. And when we are putting virus in a plate, we will actually see plaques, just like the viral plaques. All viruses won't make plaques. Plaques is just like colonies. In colonies, we are actually seeing the growing mass of microorganism or cells. But in plaques, it's actually a clearing zone of virus. If we are infecting a monolayer of cell, it will kill surrounding cells. So that's what a plaque is. So in plaque incubation assay, you are taking a cell and adding, along with that you are adding the virus. In this example, they are actually using the chikungunya virus. Okay, so the chikungunya virus is infecting the cell, it will cause plaques. You can see the dots here. All these dots are indicating the focal cell death. Cells are dead there. So in order to prevent the spread of the virus, we are actually making a soft agar on top of this virus, so it cannot be spread into the other areas, but it can actually spread in the adjacent area. And in third case, what we are done is we are putting the cells virus along with our compound of interest in suitable dilution. Okay, and if we are putting our compound along with the virus, here you can see the number of plaques is less when compared to this one. This one has cells and virus only. Here we have cells, virus, and our natural compound. This natural compound is actually reduced the number of plaques. That means it is inhibiting the bacterial growth. In that way, we can actually screen. If you have 100 different compounds, you can actually test this 100 different compounds one by one to see that whether it is having some antiviral activity or not. So the number of plaques is actually measured as plaque forming unit or PFU, just like our CFU. Here we have PFU, and 50% of reduction in PFU is measured as the PRNT 50 value, which is the concentration of the compound required for the 50% reduction in the plaque number. Okay, in this study we are actually studying the cytotoxicity also. Next method for antiviral compound bioprospecting is inhibition of viral induced CPE. CPE is actually cytopathic effect. Not all viruses is killing the cells. Some of won't kill the cell, but it will have some structural changes in the cell, just like this. This is the control cell. It's a different types of cell. That's why it's look different than the fibroblast that we have seen earlier. So this is the monolayer. Once the virus get infected, the whole cells going to round up, but it won't die. Okay. Sometimes it eventually die, but it won't lead to any immediate death. So your virus is actually causing a structural change in the cells. So if you want to test the antiviral activities, what we have to do, along this virus you need to add the natural compound, the compound you want to test. If the compound is able to prevent this type of change, it has some antivirus activity. So CPE induces the rounding of cells or fusion of cells with adjacent cells or appearance of the inclusion bodies in the cytoplasm and nucleus. So what we do, we add our antiviral agents. So if you add the antiviral agents, this is just an example. If you are adding antiviral agents, viruses and cells, if you are seeing something like this, it means that this compound is actually inhibiting the virus. Actually, if you are adding virus, it should be look like this. But when we are adding this potential antiviral compound along with this, it actually reduced the structural changes. CPE is reduced, so it has some effect. That's way we can actually find the antiviral agents from nature. Then next test is virus yield reduction test. It is a two-step procedure. First, what we do, we will infect the monoviral cells with the virus and the potential compound, and then we'll do a qualitative assay to see that how much virus incubation has been done. It's a quantitative method, it is a labor intensive method, and it is one of the most effective method to see the real antiviral property of a compound. Here is how we are doing it. It's an example of Zika virus. We have the plate which has having virus and antibacterial compound along with the cells, and after infecting with the cell we are incubating it, and after incubation we are actually identifying the number of viruses, quantifying the number of viruses using RT-PCR, qRT-PCR techniques. So if the number of viruses is reducing, that means your compound is having some effect. If it is not reduced, that means your compound is not that good. So that's how we are actually bioprospecting the antiviral agents. Thank you so much.